Hi everyone, welcome to week 4, chapter 4, how cells obtain energy. You'll notice throughout this presentation that there are going to be some extra slides included in this presentation that are not in your textbook. I find that they help explain what's in your textbook a little bit clearer, either by illustration or by some definitions that might not be in your textbook. So don't get confused. I will go page by page as always, but I will be throwing in some extra slides like this next slide just to help you out a little bit. Okay, let's get started. As I mentioned, this slide is not in your textbook, but I feel you need to have some definitions before we move forward. So before we can talk about energy, we need to talk about one more type of classification of organisms. We classify organisms into autotrophs and heterotrophs. And autotrophs are organisms that will make their own food based on energy coming directly from the sun. So they are essentially taking sunlight or visible light and turning it into chemical energy. So sun energy into chemical energy. Those are autotrophs. Heterotrophs are organisms that cannot make their own energy, and the only way they can gain energy is by consuming autotrophs or other organisms that have consumed autotrophs. Autotrophs will make their energy from the sun by way of photosynthesis. Now, I'm sure you've all heard that before, but we're going to get into the nitty-gritty details of photosynthesis in just a few slides here. But for right now, photosynthesis is just a process that autotrophs use to convert sunlight into chemical energy. This slide is a really good depiction of how heterotrophs and autotrophs utilize energy. And we're going to have another series of classification names here. Producers are just another name for autotrophs, and consumers are another name for heterotrophs. Okay, so we've got this new classification system. Oftentimes you'll just hear it called producers and consumers. Sometimes you'll hear it called autotrophs and heterotrophs. And sometimes you'll even hear it called producers, consumers, and decomposers. And we'll talk about all three of those in just a second. So here's a picture of energy coming from sunlight. In the form of heat, it reaches producers. Producers will take in that heat and that visible light energy, and by way of photosynthesis will convert it to chemical energy in the form of glucose and utilize it for various processes throughout the body. Consumers, as we noted previously, cannot make their own energy. They cannot take energy from the sun and convert it into usable energy. The only thing they gain from the sun is warmth. So in order to gain energy, we have to, as consumers, which humans are, humans and other organisms that are consumers, will have to consume producers, and producers are plants. Anything green, basically, any green plant material. The sunlight will come in to specific types of cells that we'll talk about later and reach a pigment called chlorophyll, which I'm sure you've heard before. That gives the plant its green color. And so plants will take this energy in, it will convert it using photosynthesis and make it into chemical energy. and will store it in the bonds of their cellular makeup. Remember, cells are atoms that have been combined into molecules, and molecules that have been combined into cells, right? And then those will, of course, will go on to make tissues and organ systems, and then whole organisms. Remember, at each grouping of those cells and molecules, we have bonds. We have those covalent bonds and we have ionic bonds, and these bonds will store energy. The energy is released when those bonds are broken, and again, we will talk about that later. So moving on to consumers can only gain their energy by consuming plant material. So think of producers as plants and consumers as carnivores or herbivores or omnivores. Okay, so let's start with herbivores. You may have heard these terms before. Herbivores are animals that only eat plant material. So herbivores are gaining the most energy because they are only one step away from energy from the sun. So the energy comes in from the sun to the plants, and the plants make the energy, and then herbivores will consume that plant material that has the energy. Carnivores are not going to be eating plant material, so they are not getting this mass dose of energy from plants. They can only get their energy from other animals that they eat, primarily herbivores. And so carnivores will eat herbivores or even other carnivores and get their energy from those animals that have eaten plant material. And so if you think of it as a straight line, you'll have energy from the sun going straight to the producers, going straight to the herbivores, going straight to the carnivores. And then off to the side you might place the omnivores. And the omnivores are those animals like humans that will consume not only animals, but they will consume plants as well. And so if you are a vegetarian as a human, you are gaining more energy than someone that only eats primarily meat. Okay, so you have to think of it in a who's getting the most energy, and then what am I eating, and how far removed is that from a plant material. And so let's say I, as a human, will consume a cow, and a cow will consume plant material, and of course the plant material will get energy from the sun. And so I'm only like 2 steps away from energy from the sun, so I'm getting a good dose of energy. If I am a human that, let's say I ate a wolf, and the wolf ate a rabbit, and the rabbit ate some lettuce. Well, now we've got an extra step in there. Now I'm even further removed from energy from the sun. The plant material, the lettuce, got the energy from the sun, and then the rabbit ate that, and then the wolf ate the rabbit, and then I ate the wolf. And so that's almost 4 steps away from energy from the sun. So nutrition, you can see how your nutrition is a big deal in your bodily systems, because your body can't function unless it has the appropriate amount of energy. So if all you're consuming is meat material, you're going to be getting some energy, but not as much as if you were including plant material in your diet. Now, every time energy is transferred from one organism to the next, because that's how we get our energy. Again, we're consuming to get energy. Every organism that consumes another that has at some point consumed plant material that got energy from the sun, every step in that process from animal to animal is not getting 100% energy. Every step actually loses some energy to the atmosphere in the form of heat. This is one way where our environment can gain heat. So energy that goes from the sun to the producers did not gain 100% heat, energy. It gained 90% energy, and 10% was released to the atmosphere in the form of heat. And so again, we have this stepwise situation where energy that goes from the sun to the producers is losing 10% heat. From producers to consumers, we're losing 10 more percent heat. And then from other consumers to other consumers, we're losing more heat. So it just depends on how many steps in the process there are. By the time you get your energy from eating an animal, you could have a very small amount of energy that you're gaining. So again, if you are an herbivore, you're going to be gaining the most energy over all the other organisms. Now we've got this little grouping here that I briefly mentioned called decomposers. And decomposers are organisms like fungi that will consume detritus or dead material or dying or decaying material. Okay, and so when plants and animals, producers and consumers, die, decomposers will be able to feed upon these organisms and gain their energy. And again, some is lost to the atmosphere as heat. When decomposers die, or if they don't consume all of the dead matter, whatever's remaining along with dead decomposers, producers will take up in the form of nutrients from whatever was left in the soil. So if you have an animal that died, decomposers came and consumed all over it, and there was a little bit of carcass left, and then the decomposers died, let's say, in the same spot. All of that material on the ground, in the dirt, let's say, will go into the soil, and the plant roots will take up nutrients from there. So we get this cyclical nutrient cycle happening. So nutrients cycle in a circle, and energy is really said to go from in a straight line from, even though it kind of shows it here in a circle, energy goes from the sun to producers to consumers to decomposers, and then is released, whatever's remaining, back into the atmosphere. This slide gives a little bit of an overview of metabolic pathways. Metabolic pathways are a series of enzyme-mediated reactions by which cells build, remodel, or break down an organic molecule. And they can do this two ways, either by taking in energy or getting rid of energy. And so the first section here, the first picture that you see, are anabolic pathways. These are small molecules that are built into larger ones using energy. So we're taking in energy. These could be said to be endergonic processes. So we've got several individual molecules plus energy yielding one big molecule. And that's often how we see our chemical reactions depicted in this class. We have the reactants on the left plus energy yielding the products on the right. The catabolic pathways are those that make energy. So these would be energy releasing compounds, or not compounds, but processes, and can be said to be exergonic. And so these are large molecules that are broken down into smaller ones, and by doing so, the bonds that hold those pieces together are being broken, and then energy is being released. And overall, this can be said to be metabolism, or components of metabolism. And metabolism can be defined by the complex of physical and chemical processes occurring within a living cell or organism that are necessary for the maintenance of life. And I think we're pretty much all familiar with this process. And of course, if you have a nutrition class, you'll be talking about this in depth. If you've taken physics, you've heard of the laws of thermodynamics. And in physics, they probably talk about all the laws of thermodynamics. I believe there are four. We are only going to be concerned with the first two. The first law of thermodynamics says that energy in the universe is constant and conserved. This means that the same amount of energy has always been and always will be. And an easy way to think of it, and the way you normally hear it, is that energy cannot be created or destroyed. So energy is one of those tricky little things that physicists have not gotten a complete, full grasp of what energy is. It's still abstract to us in many ways. Yes, we have these laws that we were able to discern from our studies of energy, but we don't know where it originally came from. We just know that it's always been. It's always been here, and it cannot be changed. Well, I take that back. It can be changed come in many forms. However, we can't create it, and we cannot destroy it completely. We can heat it so that, let's say if you have water molecules and you heat them, the heat is a form of energy, and it will make the water boil, and it will dissipate in a form of gas, and along with it, energy will be released. But the energy will go into the universe and be taken up and used as heat for the overall environment. So it's this one of those, it's a little bit abstract, but it's this idea that energy cannot be created or destroyed. And the second law of thermodynamics that we concern ourselves with is that every time there's a transfer of heat, some of that energy is lost to the environment. And that roughly pans out to be about 10% with every transfer. Sometimes you'll hear less, sometimes you'll hear more. It just depends on the textbook that you're talking about, but on average it's about 10% that is lost to the universe. So these are your two laws of thermodynamics that we will be concerned with in this chapter. This figure just roughly shows you some brief examples of different types of energy transformations. So we've talked about light energy coming from the sun, going into plants and being converted into chemical energy. Chemical energy can then be used to form other molecules that, let's say, lipids, that would be the fat content, let's say, of this ice cream. The bonds of those molecules you'll gain energy from once those bonds are broken. So once you've licked at the ice cream cone, you've broken a few bonds and you've gained a minuscule amount of energy from your ice cream. This amount of energy that you're receiving from food can be transformed into kinetic energy. Kinetic energy is the energy of motion. Okay, and so of course you'll talk more about this in nutrition, but depending on the type of food you're eating will depend on how much energy you can gain from it and how much kinetic energy that can be transformed into. And so ice cream probably isn't going to give you a lot of long-term energy. It might give you a short burst of energy to maybe get your little bicycle moving there. But let's say maybe 5 or 10 minutes of pedaling the bicycle, you might run out of energy or steam because that one ice cream cone you ate really isn't enough to sustain you as far as energy or nutrition for that matter. Before we can talk about this form of energy called activation energy, I want to give you some examples of what chemical reactions look like. And so in this picture here, I have a chemical reaction listed for you. This is actually the molecules that make up water, and then using energy they combine and form water. So on the left we have two H2 molecules. Okay, so we have two hydrogen molecules plus one oxygen molecule. When we combine those two and then add a little bit of energy to them, we end up with two water molecules, or 2H2O. And the picture can show you this a little bit better in a visual. So you've got two hydrogen atoms combined, and then you've got two of those, which is where that big number 2 before the H comes from. And there's two hydrogen molecules combining, and that's what the subscript 2 next to the right of the H means. So we've got two hydrogen molecules that are formed, and we've got two of those. And then it shows you've got two of the oxygen molecules combining. And so by way of chemical bonding and hydrogen bonding, you end up with two water molecules. So one oxygen molecule combines with two of the hydrogen molecules, and then that will happen again. One oxygen molecule combines with the other two remaining hydrogen molecules to form two water molecules. Now, each part in a chemical reaction has a name, of course, right? So we wouldn't have science without naming stuff. So the components of a chemical reaction that enter into the reaction, so the ones that are being added together, the hydrogen and the oxygen in this case, those are called reactants because they are going to react together and they're going to react with energy. Okay, the end result in a chemical reaction. Usually the component to the right of an arrow, as it's written at the top there, those are going to be called products. So in this case, we have two products forming. We have two water molecules that are your products. The triangle before the E above the arrow there, the triangle means a change. Okay, it's a mathematical symbol, and so we use it a lot in chemistry and physics and science of all kinds. So we've got the change in energy combining with the hydrogen and the oxygen to make two water molecules. So we've got two reactants plus energy combining to make products. Now it's important that you understand those terms. You may also see, especially if you had chemistry before, that chemical reactions can often go the other way as well. You can start with a product on the right, the arrow would be flipped around facing to the left, and you would end up with the oxygen and the hydrogen. Okay, but for now we're just going to go left to right. Again, I want to give you one more piece of background information before we really delve into these energetic equations, chemical equations. Illustrated here is just a nice little depiction of how much ATP is in the human body. And ATP, if you've not heard of it before, is a form of energy, and it's the most typical kind of energy that our body uses for all sorts of chemical processes in our body. ATP stands for adenosine triphosphate, which means it has three phosphate groups. The tri means three, the P means phosphate, and so this is saying that there are three of them. And the very first sentence there, ATP is enzymatically reduced to ADP plus PI. What happens sometimes, and it's illustrated here, is that ATP will be used, and in order to be used, one of the phosphate groups needs to be removed. And so you end up with removing the one P, which in this case is called PI, which means inorganic phosphate. So you take out that P and you're left with a DP, D as in dog, for adenine diphosphate. DI means 2, the number 2. So we have 2 phosphate groups left, plus a phosphate group left over from that. So ATP can be enzymatically reduced to ADP plus an extra P, which is inorganic phosphate. You can also go the other way and convert the two back to ATP. So you can combine ADP plus PI using enzymes and get back your complete ATP. And so this is just showing you that every ATP molecule will cycle through this reaction about 5,000 times every 24 hours. And so there's a big long number there. I'm not even going to try to say. That's 120 with all those zeros of molecules created in the human body. So clearly that's a crap load. And notice that the arrow goes both ways. ATP to the right, ADP to the left. Now we can sort of put these ideas together. Remember, we're talking about cells taking in chemical energy, or excuse me, taking in light energy and converting it to chemical energy, storing that energy in the bonds, molecular bonds, and basically you're saving it for later use, because we don't use all of our energy in our body all at once. Not all chemical processes in the body happen all at once, and so some of the energy needs to be stored for later use. Okay, so this says cells store and retrieve energy by making and breaking those chemical bonds in metabolic reactions. Some reactions require a net input of energy, which is sort of what we were talking about before. This is a better illustration of that. We have our reactants being added together, and then we're taking in energy in the form of ATP, and we're creating this product of ADP and the PI. And so when we take in energy into a chemical process, it's called an endergonic reaction, which is just a long, complicated word to mean that we are taking in energy. Energy is going in to the actual chemical reaction. Here we have the flip side of that other reaction. This is other reactions that when they are all said and done, there's a net release of energy. So we're taking reactants of the ADP plus the PI, and we are releasing energy to form a product. And at the same time, we're forming ATP. And this is called an exergonic reaction. So this is the energy exiting the reaction. It's a nice way to say energy out. Okay, so before we had endergonic reaction where we had energy in, and this is an exergonic reaction where we have energy out. Here's one more way to just sort of tie up this notion that we've been talking about. So energy in and energy out is basically what we're talking about here. Chemical bonds hold energy. Cells will store energy in the chemical bonds by running energy requiring reactions, and those are called endergonic reactions. So we're having energy in because we are requiring energy to come in and help us make these products. Cells access energy by running energy releasing reactions, and these are called exergonic reactions. So we need energy to exit the reaction in order to form our products. Energy in and energy out. I mentioned activation energy earlier. And simply put, activation energy is the minimum amount of energy required to get a chemical reaction started. So let's say we had a chemical reaction and we threw a whole bunch of energy into it in an endergonic fashion. So we're taking energy in, and let's say this really revved up the chemical reaction and it got started super fast, and the reaction was well on its way, and halfway through the entire reaction we're all excited because we're going to end up with these great end products. However, halfway through, the reactants burn up, and therefore we don't have any products left, because without reactants we can't combine them. We have nothing to combine to make products, right? And so Energy is this very forceful, powerful thing. And we don't always need that much energy to put into a reaction to get our end result. So activation energy is just a little bit of energy required just to give you a little nudge to get that reaction started, and it'll get the ball rolling, and your reactants will combine along with that energy, and you'll end up with your nice products at the end. And so that's what activation energy is. And this particular slide is combining our next topic, enzymes. And when enzymes are exposed to activation energy, or actually it's the other way around, activation energy is exposed to enzymes, the enzymes will actually lower the overall energy of the reaction. Okay. So this is showing you that it's got a reaction path, which is basically the reactants mixing along with the energy, and then you get these end products. If we didn't have enzymes, then there might be too much energy involved in this chemical reaction, which is what that purple line is showing you, how the hump, if you will, goes way up at the top and then comes down. If we use an enzyme, it lowers that activation energy, and so our reactants start out at a lower, slower form and can tootle along the way very nice and slowly at a consistent rate, but at a much slower rate. And it will slowly climb this hill, make a little bit of a hill, and then come back down. And when we're all done at the bottom of the hill, we have these nice products that we were looking for. So activation energy is a little bit of energy required to get the chemical reaction started, and enzymes will lower that activation energy to make sure that nothing's burning up too quickly. Okay, before I get into this whole picture, you'll notice that there are words in here, especially where the green half circle and triangle are in the upper left corner here. Those are being called substrates. Well, if you go back to the image of the chemical reaction where we had one reactant plus another reactant will eventually make products. Well, when an enzyme is involved in a reaction, the reactants are then termed substrates. And it's not to confuse you or throw you one more term, which I agree can be confusing. It's just another identifier for you to say, oh, substrate. I know that that is a reactant plus another reactant using enzymes to make these products. So whenever I see the word substrate, oh, those are reactants that are using enzymes to get the end products. So here we have these substrates, or they can be called reactants, but the technical term for these are substrates because we're going to be connecting them to these enzymes here. The enzymes are the gray half circular sort of weird looking Ms. Pac-Man, as I like to think of it. And again, yes, I'm showing my age. However, you can see that we have this nice half circle and triangular green substrates. The red arrow is showing that they need to go connected to this enzyme at this surface area. The surface area that it's pointing to is called the active site. The active site is the area on an enzyme that accepts the substrates, or that connects the substrates to the enzyme. Now you'll notice that that half circle is a little bit taller in depth than the circular half circular area on the top of that active site. It's not quite as deep. It's pretty shallow. Well, this is where this induced fit model comes in. It was previously thought that enzymes and substrates had a lock and key mechanism, where enzymes were the exact shape, the active site on an enzyme was the exact shape of a substrate. As technology has improved, we've been able to determine that that is not quite the case. It does eventually get this lock and key mechanism happening, but before that, we have enzymes where the active sites are sort of this fluid, motional, having motion surface, if you will. And so the active site may start looking like this, where you think, oh, that's not going to quite fit, but this particular substrate will come up to this particular enzyme and it will land on it, and as it does, the enzyme will form to make that lock and key mechanism happen. So every enzyme active site is made for one specific type of substrate. So if these were two half circles on the substrates instead of a half circle and a triangle, and they came up to this particular active site on this enzyme, the enzyme would not form to meet it to make a lock and key mechanism. Only this particular active site will accept this particular substrate. So a half circle and a triangle will land on this active site on the enzyme. The enzyme will morph in such a way that it will nicely curve around and make a deeper indentation to fit that deeper half circle and make a sharper triangular area for the triangle. And now in that second picture there, you've got a nice lock and key mechanism happening. Once those are locked in, the substrates can now be altered by the enzyme into products, and they're very specific products. Again, we're looking for specific end products, and so once they turn into the specific products we need, the blue half circle and the yellow triangle, the enzyme will release them and they will go on their merry way to do whatever it is they're going to do. And then you'll notice that the enzyme on the very end will go back to its original shape, and it can work again and again and again and again, millions and billions of times over. They are not burnt up in the reaction because they are not necessarily taken into the chemical reaction. They sort of, I like to think of it as they bounce into the reaction, hit those substrates, use the energy in the manner that it needs to, change these products, let the products go, and then they bounce back out. They're not fully taken in like energy is. Energy has an endergonic process where enzymes do not. They come in, they do their job, and they get out. Okay, and then they are able to be used again and again. So, induced fit model says that we have a specific enzyme combining with a specific substrate to form specific products. Once the products are formed, they're released, and the enzyme can be used another time or a million times. This slide is a variation on the previous one. Here we still have an enzyme, again a very specific one. They've sort of tilted it on its end so that the active site is actually on the left-hand side of the enzyme instead of on the top. So the enzyme is still the gray part. The active site is that half circular area on the left, and then we have on the back side this thing called the allosteric site. Again, it's another weird word, but it's just one of those you're going to have to remember. An allosteric site, and then it's pointing to an inhibitor, and then the other picture we have almost the same picture except for we have an activator. Those little orange squares will change names depending on what we're doing. So let's take this picture on the left. This is called the allosteric inhibition diagram. So again, we've got the gray enzyme with the active site on the left. We have the allosteric site on the lower right. And then here comes a green substrate, that half circle, that we expect to have attachment to the enzyme. But what happens is, because this is an inhibitor that's coming in, that orange square is an inhibitor, it's going to lock into that allosteric site, and that nice half moon circle that the substrate normally would fit in all of a sudden changes shape just at the right moment that the substrate's going to come in to try to connect. And because the inhibitor is locked in place, it instantly changes the shape of the enzyme, and the substrate will bounce off of that active site because it can no longer fit there, clearly, as you can see. So that is called an allosteric inhibition model. On the right, we have the allosteric activation model. And so not all enzymes, basically what both of these are illustrating is, not all enzymes are just sitting there waiting for something to come and attach it to. Enzymes move around, and sometimes we require them to be active, and sometimes we require them to not be active. And so that first picture that we just talked about, the allosteric inhibition, sometimes we don't want our chemical processes to be happening all the time. And so they need to be turned on, if you will, or turned off, if you will. And even though there's not really a switch turning these on and off, there is this mechanism that's altering the enzyme shape so that the lock and key method can't happen. And think of it like this. If you had insulin oozing out of your system all the time, you have the potential for your blood sugar to lower quite low. Well, in order to combat that, we need to turn it off for a little while. So we need to turn off the insulin, and this is not an exact example. This is just a brief example. I'm not giving you all the details of how insulin works, but the inhibitor will lock in there, keep the substrate from connecting, and so that chemical process of releasing the insulin will stop. So insulin will not be released, and so your blood sugar has a chance to rise to a normal level. If you're a healthy individual, then when you eat, it'll probably rise a little more, and then you might have a situation where you have this allosteric activation happening, where the activation is now saying, oh, we're ready for you, insulin, go ahead and release, because we've eaten too much sugar or too much food or whatever it is, and our blood sugar is getting a little too high, so we need the insulin to be released now so that we can lower that blood sugar. So it works the same manner. You still have this activator, which is still depicted as an orange square. It's still going into the allosteric site, but notice we have the little tiny divot on the active site on the left hand side. It's not that nice big semicircle. So when the activator locks in there, the enzyme will change and make that nice half moon shape so that the substrate can lock in there perfectly at the activation site. It'll fit, it'll make the products, and then it will release the products, and then it'll go back to the previous shape. Okay, and so that's how sometimes we want chemical reactions to happen. Sometimes we want them all the time. Sometimes we want them just here and there, and sometimes we don't want them at all. So it just depends on what we are talking about. But we have these inhibitors and activators that work with enzymes that keep substrates from attaching, or they allow them to attach in a specific manner to make the end products that we are desired. Okay, this picture can be a little tricky. But it sort of builds on the idea that we were just talking about with the allosteric site and the inhibitor molecule attaching to an enzyme. If we start to the far left with the enzyme there on the left, the gray little Pac-Man looking figure, and that green substrate triangle, let's start there, and you can see that the substrate will attach to that first enzyme, which is the one depicted. And then there's going to be this intermediate substrate along the way, along the path of some chemical reactions. So they don't necessarily happen in this nice straight line that we see, but a lot of times they will happen if you imagine a straight line and the substrate moving through that straight line. Along the way, it's going to be gathering up other components. And so we're going to pick up an enzyme, we're going to pick up these substrates from all over the place. Okay. And so it's not always just a straight line, but it's easiest to think about it like that. So we have the substrate going along the way, and it attaches to enzyme 1. And then it will utilize this first intermediate substrate, which basically is the end product of this chemical reaction. And that substrate will now attach to the enzyme 2, which will change it into the blue ribbon looking thing, if you want to call it that, which is the second intermediate substrate. And then that will go to a third enzyme, and notice that each one of these enzymes are shaped specifically to meet that particular type of enzyme. Okay, and so we have got this blue enzyme attached to enzyme number 3, and we have this final end product, which is this little yellow square. Well, the yellow square then actually becomes an inhibitor molecule, and it will attach to an enzyme like we first encountered in enzyme 1. And the substrates, there's not just one in the body, let's say. There are thousands or millions or even billions of them, okay? And so they are attracted to the enzymes. And as we said before, they will not fit all enzymes, but this particular scenario shows a green triangle should fit into this enzyme 1. Well, a green triangle comes up to enzyme 1 down here now at the bottom left, but it can no longer attach to it because we have this yellow square that has become an inhibitor molecule on the allosteric site, and it has changed the shape of enzyme 1 so that the substrate has bounced off of it, and it will go along its merry way, and the molecule, the inhibitory molecule, can be released from the allosteric site, and the enzyme can go back to a triangular shape, thus accepting a new substrate that fits in there, and starts this whole process over again. Okay, so then it goes to enzyme 1, to enzyme 2, enzyme 3, and uses these intermediate substrates along the way. And so this whole mechanism is called feedback inhibition. And so enzymes can be a very tricky business, especially once we start talking in this realm. It's not just, hey, there's a triangle, goes up to this nice enzyme that has a triangular shape, and we make a nice end product, and then we go again and again and again. Well, that's not always the case. As you know, the body is a very complex thing, and so are every little chemical mechanism involved. And so this is one of those illustrations that show just how complicated it can be. But take it frame by frame and really think about what we've said before in the previous illustrations, and you can have a good understanding when you look at this picture of what's happening. Here's another example of the feedback inhibition. If you're not such a linear thinker, this one is a little more circular in nature, but it depicts pretty much the same thing. So if we start, and I want to note that they're using the term here straight in the middle at the top of this circular picture, reactant. Well, we know that if enzymes are involved in a chemical reaction, the reactant is going to now be called, fill in the word here, yes, substrate. I know that's what you all said, right? So pretend that reactant says substrate. And the substrate again will come up to this enzyme, it will attach to it, it will create a product that will become this intermediate helper, if you will, intermediate substrate, and that will attach to enzyme number 2, which will put off a third kind of substrate or product. And if you take the inner circle, you'll hit enzyme 3, and your end product is going to be all these different types of products. They might be the same product, but there's going to be a bunch of them, is what I meant to say. Or from enzyme 2, the product that's formed could actually become another intermediate and go back to being the substrate, or as it says here, reactant. Okay, so this one's a little bit more circular in nature to think about. I think I'm a little bit more linear in thinking, so this one is a little more difficult for my brain, but it's basically the same thing. You have a reactant, but because we're using enzymes, we're going to call it a substrate. The enzymes are going to create these intermediate substrates that will attach onto other enzymes, and you'll have this chain reaction happening. At the end, you'll either have products, or you will continue on in the cyclical path. Your substrates will go on to do this again and again and again. And hopefully the end product will, that hopefully you will have an end product by utilizing from enzyme 2 to enzyme 3, having those products in the middle. Sometimes they just go around and around and around and around. But eventually, hopefully you get that product that you're looking for. I mentioned this earlier, but this is a great illustration of the chemical compound for ATP. Remember, ATP, whenever you hear the words or the letters ATP, automatically think energy. It is energy. ATP is the main form of energy that our body uses for chemical processes to carry out different functions in the body. Okay. And we'll talk about exactly how that's going to work in just a little while, but this is just an illustration of what the chemical compound of ATP looks like. On the left, in the 3 ovals, inside each oval you have a phosphate group. Phosphate group contains phosphate and 3 oxygen molecules, and you have 3 of those, so you have triphosphates, right? And then the rest of it would be your adenosine, or adenine in this case, because you're going to use adenine. And then you're going to use a ribose, which is a 5 carbon sugar, and mixed with these phosphate groups. So if you have 3 phosphate groups, it's triphosphate. If you have 2 of them, it's diphosphate. And in some cases, you may even hear one phosphate group, and you'll hear the word monophosphate. And so I just wanted to show you what that chemical structure looked like to give you a little bit better visual of what we're talking about when we say ATP. And then when we break it up into ADP and a P. So if we just had two of these, that third one would be off by itself, and that would just be the P, or phosphate. So basically, glycolysis is sort of a byproduct of photosynthesis. Um, you can see here at the top we've got this six-ringed glucose molecule, and through some energy happening here, the glucose molecule will break apart basically and be utilized, releasing energy, and the energy again is in the form of ATP, and here we're releasing two ATP molecules, and then some ADP have been produced as well. Once that energy, the ATP and the ADP have been taken apart or taken out of this glucose ring, you're left with this stuff called fructose diphosphate. Remember, now we've got two phosphate molecules here. Each one of those will break up into these, it's called the glyceraldehyde triphosphate or 3-phosphate, and you'll have two of these, and now you can see they're on very separate paths. So you've got the left hand side and the right hand side, but each side are producing the same things. Once it goes through these chemical processes using coenzymes and cofactors, you've got NAD plus NADH being extracted. You've got more ADP and more ATP being extracted, and then your end product is what's called the pyruvate. Okay, and then you've got each side giving off both of those, right? And the energy and the coenzymes. So basically, if you count up the ATP, we've got essentially 4 being produced by glycolysis. So we've got 4 energy molecules being produced. This picture is showing how the pyruvate from the breakdown or byproducts of glycolysis in the previous picture that we had can be utilized with some coenzymes of the coenzyme A and the NADH and NAD plus can form this acetyl-CoA. And again, using a coenzyme, it enters into this citric acid cycle. If you're older like me, you've probably heard this called the Krebs cycle, or even other textbooks that you have might say the Krebs cycle. This is the same thing. So citric acid cycle and Krebs cycle are the same thing. Once the acetyl-CoA enter into the citric acid cycle, a series of coenzymes are used and produced, and you'll see on the as we go around the upper part towards the right of the circle here, we're going to be giving off some carbon dioxide molecules, and then we'll still continue within the citric acid cycle. We're going to use ADP to form some energy to put off ATP, and then we will keep going around the citric acid cycle, and it'll go around and around. It's called a closed loop, and so that means that the last part of the pathway will regenerate the compound that will be used in the first step of the pathway. So it goes around and around and around, and it says here that the 8 steps of the cycle are a series of chemical reactions that produce 2 carbon dioxide molecules, which are shown there on the far right, and one ATP molecule, which is shown down below on the bottom part of that circle. And then the reduced forms of NADH and FADH2 that came from the NAD plus and the FAD are coenzymes that are important that help this process along. Okay, and again, electron pathway system is going to be utilized, which we will talk about in the next slide. But basically, this 2 carbon atoms will come into the citric acid cycle from the acetyl group, and in this case, there were the pyruvate acid from the glycolysis is making this acetyl-CoA. And then they will eventually be released in later turns of the circle, making those 6 carbon atoms from the original glucose molecule. Okay, and it says here that it takes 2 turns of the cycle to process the equivalent of 1 glucose molecule. And then also we have some coenzymes happening here as well. But mostly the thing you need to know is that this entire process, the pyruvate all the way down into the citric acid cycle, this all happens within the mitochondria of the cell. So if we went back a slide, we would see that the glycolysis happens in the cytoplasm, and then the Krebs cycle or the citric acid cycle process happens in the mitochondria. At the end of the citric acid process or citric acid cycle, we are producing 6 carbon dioxide molecules and some ATP. Okay. This picture is in your textbook. It's figure 4.15. I find it difficult to read, and maybe it's my older eyes, but I think it's a little bit blurry. And again, that might just be my eyes, and I find it really hard to read on this slide. And so if you can read it, great. Go back and read the description. Basically, this is the electron transport chain that we're talking about. You can see the citric acid cycle in the circle in the lower picture. And basically, it's a set of molecules that will support a series of oxidation reduction reactions. And then ATP synthase is a complex molecular machine that uses a hydrogen gradient to regenerate ATP from ADP. Basically, what I want you to get out of this is the use of the electron transport chain. All right, this is the free energy from a series of reactions and is used to pump hydrogen ions across a membrane. And then basically we are slowing down a reaction. If you look at the next slide, I think this picture will make a little bit more sense in how an electron transport chain works. Okay, here is a depiction of an electron transport chain, which I think makes a little bit more sense. Mostly I want you to focus on the little stair step thing happening here, but we'll back up a little bit and we'll start with glucose. Glucose is going to utilize an enzyme to get rid of carbon dioxide and some hydrogen ions. The hydrogen ions will help form water at the end of the staircase thing. But the glucose will, once the CO2 and the hydrogen are removed, you're going to have electrons that are going to enter into this transport chain. And so electrons and enzymes are going to work together in a stair-step fashion to lower the overall amount of energy that is in this process. And so energy will be released, but in small increments as we go down the staircase. And the whole point to this is if we didn't have this electron transport chain, the energy involved in this process would be moving so fast and so quickly that the energy would burn up the reactants before it could make the end product. And we've talked about that before. So we need to have enzymes for one, helping along with the electrons, slowing down this mechanism in a stair-step fashion. And we say stair-step fashion because we're doing small increments at a time. We're really utilizing the electrons and enzymes to release a small amount of energy here and there to get that end product of water. And then it starts all over again, okay? But the stair stepping down is important because we don't want that reaction to burn up before it's had a chance to give us the products that we need. So in aerobic respiration, we have this component to the process. So we're going to have the electron transport chain slowing down in small increments the amount of energy that's given off. And in this particular example, water is created as a byproduct. Here's a nice way to think about what we've just talked about. We've thrown a lot of information at you. Aerobic respiration starts in the cytoplasm, and it's called glycolysis, which is stage 1. There are reactions in which glucose or another sugar is broken down into 2 pyruvates and will release 2 ATP molecules. Coenzymes, in this case 2 NADH molecules or enzymes, will pick up the electrons and then transfer them into the citric acid cycle or the Krebs cycle, however you want to think about that. Those electrons are moved into the mitochondria, and that's where this citric acid cycle takes place. So we've got acetyl-CoA formation that's come from the glycolysis stage, from the pyruvates basically in the glycolysis stage, and we've formed this acetyl-CoA, and then we've broken down pyruvate to carbon dioxide. We've netted 2 more ATP, and we've reduced coenzymes. Now we have 8 NADH and 2 FADH2. There's a note here that says the text on page 90 incorrectly shows 6 NADH. That is actually for a different textbook, not the one that you're working on, not the textbook called Concepts of Biology for the Bio 156 class. So just ignore that little note. There are 8 NADH and 2 FADH coenzymes produced, okay? And all of this occurs in the mitochondria. So glycolysis occurs in the cytoplasm, and it starts with glucose. It ends up with 2 pyruvate, and then the pyruvate along with the electrons are going to cross into the mitochondria and start the citric acid cycle, which will take the pyruvate and utilize an electron transport chain to get the acetyl-CoA formation, and you're going to be giving off CO2. You're going to be gaining 2 forms of energy in the form of ATP, excuse me, and then you're going to have these coenzymes as well. And again, that occurs in the mitochondria. So these are the most important facts about aerobic respiration that I can share with you. Here's a picture I really like. It's not in your textbook, but I think it gives a really nice visual of what's happening in the aerobic respiration process. So if you look at this picture, the top part shows you where glycolysis happens. If you'll notice the white area, if you will, picture that to be the cytoplasm inside the plant cell, right? All right, so we've got the cytoplasm. We have 2 molecules of ATP and 2 NAD plus entering into, along with glucose, this glycolysis process. Now remember, glycolysis literally means the breaking down of glucose. So we're going to take that glucose that we got from photosynthesis, and we are going to take out some of the coenzymes. We've gone from the NAD plus to the NADH, and then we have 2 pyruvate, and then we've got a net yield of 2 ATP happening off to the side there. Then the NAD plus will enter into the mitochondria as well as the pyruvate. The NAD2 is going to meet up with the pyruvate. It's going to have 2 CO2 molecules removed, and you're going to have a product of 2 acetyl-CoA molecules. Those are going to enter the Krebs cycle along with we now have 8 NADH and 2 FADH2. This is where it should say 8 and not 6 NADH. So we have 8 NADH and 2 FADH2 entering into the Krebs cycle along with the acetyl-CoA. As we go through this process, we are going to get rid of 4 CO2 molecules. We're going to have those released, and 2 more ATP molecules. And so this whole process thus far has given us a total, along with the electron transfer phosphorylation happening, 32 ATP molecules. So we've just from 6 glucose molecules, we've netted 32 ATP molecules. That is a lot of energy, and I want you to remember that when we start talking about anaerobic respiration. Okay, and note the difference in how much ATP is being net in this case. All right. And then the next slide will explain this electron transfer chain. The word phosphorylation just means that a phosphate group is being removed or added to another phosphate group. Okay. So here we have another picture that is just being carried on. This would actually be attached to the previous picture off to the right of it. And so we kind of went from a north to south realm, from glycolysis to citric acid cycle. And now we're going to the right and entering into the final stage of aerobic respiration. And this is where the electron transfer chain that I talked about earlier happens. And again, we're utilizing hydrogen ions and these coenzymes, the NADH and the FADH2, and we have to have these electrons entering in this chain to lower the amount of energy happening so that we can have the end product of producing the ATP and all the hydrogen, and then of course the water as well. And so what this does is, once you go down this stair step of electron transfer chain, you will take the oxygen that's formed and you'll grab onto some more hydrogen that's in this membrane, and some of it will shoot across the membrane back into the cytoplasm, and the energy, the ATP, is going to be yielded and to be used later on for other processes. And that ATP also comes from the addition of the ADP and the phosphate that it picks up along the way as it crosses over. But the biggest part about this final stage is that it has an electron transfer chain that slows down the energetic process so that we can get these end products as we expect and need, and not burn up too quickly. Now let's talk about the anaerobic respiration process, often called fermentation. There are two types of fermentation: lactic acid fermentation and alcohol fermentation. And we can glean from the word anaerobic that there is no oxygen involved in this form of respiration. We are still going to be beginning in the cytoplasm with glycolysis, and exactly as we did in aerobic respiration, the exact same process of glycolysis. So we're going to get glucose, we're going to use the glycolysis process, and we're going to end up with the 2-pyruvate. Then we're going to use this fermentation process, and we're going to end up with lactate molecules. And specifically, we'll end up with 2 lactate molecules. You're probably most familiar with lactate when we call it lactic acid. And lactic acid, again, you've probably heard of in relation to working out. If you are someone that works out at the gym, particularly if you lift weights, this is a very common thing that happens with weightlifters. They will well, let me back up just a little bit. Lactate can form in the muscle. And in order to lift weights, well, of course, we use our muscles, but we also have to have some oxygen happening here. And so when we lift to the point of fatigue or the point of exhaustion, we've utilized all the oxygen in those muscle cells. And what is left is this lactic acid buildup. And so if you're a weightlifter, you know that you need to, when you've completed your sets, you have to get a good rub down, and preferably right after your workout. Any The longer that the lactate or the lactic acid is in your muscles, the longer it's going to take for the effect to go away. And that effect is a soreness of the muscles. And so if you're a bodybuilder and you've lifted to fatigue, you now have this lactic acid buildup happening. If you have not gotten a proper rub down, or none at all, the next day, or maybe even later that day, or even 2 days from the day you lifted, you will feel a soreness in your muscles until that lactic acid can be taken up and eliminated by the liver. So that is the most notable form of lactic acid that we have, and that is a type of fermentation, because the oxygen in those muscle cells has been diminished or used up, basically. And so what lactic acid's basically a byproduct of that. And then the alcohol fermentation is quite similar, and we'll talk about that in the next couple of slides. This depiction is in your book. It's just a brief way to show you how lactic acid fermentation happens. Again, you'll note that we are starting with glucose and the glycolysis process and ending up with the 2-pyruvate. That's all happening inside the cytoplasm. Then we have the fermentation happening and the two lactate molecules, and because we have this situation where the NAD plus is moving in and out of the both processes, we get this lactic acid fermentation happening. And you'll notice that there's no oxygen in this process. And you'll also notice that the NAD plus that's produced by fermentation will be the same NAD plus that is enters into the glucose and glycolysis process. This is basically how we get alcohol. This is the second type of fermentation. It's the alcohol fermentation process. And basically, pyruvic acid is going to yield carbon dioxide and acetaldehyde, and the acetaldehyde will remove the NADH and form NAD plus, and so we're getting rid of that, and then the byproduct is ethanol. And your book gives you the little nitty-gritty process that this happens, all the little details, which for this class you don't need to know, but just this particular depiction, these two chemical reactions, that's really all I care that you know. Just know that it takes from pyruvic acid, we can gain ethanol by taking out some carbon dioxide and some NADH. Okay, and that alcohol fermentation is a type of fermentation, basically. And then your book talks about and gives some examples of if the carbon dioxide produced by the reactions not vented, letting air into the chamber, then like when you're making beer and wine, then it will remain dissolved in the actual fluid. So it'll be dissolved in the beer or the wine. And then you'll have to create quite an abundance of it to get to some wine that has a lot of alcohol in it. All right, and that's where we're going to end this chapter. There's a little bit left to it, but this is where we're going to end for our purposes.